Yes. First of all, I would like to thanks for giving this great opportunity. Myself, Dr. Dharmaraj from India. I'm working as an assistant professor in zoology. Today we are talking about first time DNA barcoding of spider from the Nilgiris, Tamilnadu, India. Why I'm said like that? In my area, it's very close to Western Ghats because the last previous studies is not working about barcoding systems, but myself only the start as DNA barcoding of spiders from the Nilgiris. So the coming to the introduction, the worldwide the molecular work is very different, and then terrestrial organism is grasping of insects, and then arachnids provides an essential service keeping insect population under control. Already we know that. And then spider identification using morphological characteristics is time consuming and a hectic job for different reasons. And then DNA taxonomy, the DNA taxonomy in the last 30 years, it is an advanced work in genetic analysis. And then analysis based on DNA taxonomy have been used in studies of spider population as well as for specific delimitations. And then study the genetic structure in spiders by analyzing their mitochondrial genes sequences. Coming to the aim and objective, it is the main objective as to study the molecular phylogenetic analysis of selected species. Molecular studies of spiders is the establish a genetic references library for future study of spiders in molecular research. I am selected these kinds of area like two selected areas like Avalanche and Emerald Valley. This map is very close to Western Ghats in India. So this is my study area. So these types of two study areas like Avalanche and Emerald Valley, the both areas I have researched, and then I have concentrated in the agricultural area and then forest ecosystems. So the two selected areas like 321 square meter color, 321 square kilometer covered by Avalanche area, and the 289 square kilometer covered by Emerald Valley area. So these kinds of crops only I have selected in my studies. Like first one is tea plantation, second one is beans, and third one is potatoes in the both areas. And then very close to forest, I have selected these types of habitats like water tree plantation, grassland ecosystem, and the solar forest. So my main methodology is like the molecular analysis, like genomic DNA by using cogen DNA kit, and then 1% of acrogel for using PCR analysis, and then 2% of acrogel for using the PCR confirmation, and then sequence the sample, and then make the phylogenetic tree. I have selected these kinds of species like Archaeopanasuja, Nepalia coli, Tenagantha species, Gelata species, Posa agrinites, Archaeopus brahminicus, Archaeopus jamanans, Archaeopus feta, and then Dendroidan gitae, Carotus vidas, Monomerus pivitatus, Pelaxipus petaci, and Telemonia diametatia, like as a decorate, Holoporus species, Mymarasham melanoch parva, and the Bezoar species, Optima atlanta, Iposa, Bezorina, Neoscon species, and the Citana species, Neoscona mogaji, Archeop species, Arana species, Gastrogantha gaminata, Hernia, Artissima, Leucus decorate, Oxyad virunas, and then Hylosemicophan neocon species, Asaria sarosoni, Oxyot species, Ercelia swagna, Leucate species, Archiopus linitipus, Seraconium species, Telecote species, Thomisus species, Tostidia species, Archaeopulsella. So these kinds of species only we have observed in the agricultural areas. And then first time we are reporting in India the Acarina tabulata and then Satana species, Pintala vitata. These kinds of species were observed in the my study area. So I am using the molecular studies. The DNA barcoding has become quite popular for molecular identifications and documentation of species because it is simple, quick and affordable method. Present study was conducted to identify selected spider species such as Myrmenotium platolites, Acarina tablata, Archiopus brahminicus, and Cryptophora citricola. So these four species only I have selected because two species it is very high number in the study areas, and then two species is very low in number in the study areas. That's only I have selected these kinds of four species, but Acarina tablata it is a first time report in India. The sequences of selected spider species it is sequences with a number gene number. So the Myrmanational plotlights, it is divergence is very close to other species. And the phylogenetics of the spider species, the phylogeneity showed one cluster and a clade. First cluster was formed by Cryptophora citricola and Acarina tablata as sister taxon with a bootstrap value of 46 and Archiopus brahminicus as formed alone. The remaining species, Myrmanational plotlights, was aligned at the top of the tree. And coming to the conclusion, the main goal of our study was to explore the spider diversity of Nilgiris and compare the efficacy of DNA barcoding with morphological based evolution for species identification, and then 88% of options of diagnostic characters and availability of identification keys for juvenile spider species, and then 100% of successful results accordance with the results of Barrett at Hubbard, who correctly evaluated the 168 species of spiders using molecular tools of DNA barcoding. And then in conclusion, we can say that morphological based approaches to describe any spider species are satisfactory, but no magnify the pace or credibility of the results. Combination of DNA barcoding is beneficial. So I acknowledge to the I thanks to Bharatiyar University who has given the opportunities, and then Sri Vijay Vidyalaya College of Arts and Science, they are given the deliver to my work to your AAC virtual program. And one more thing, I also tomorrow my foreign collaborator, the Angelina Chong, also deliver the Spiders in the Spotlight, a global assessment of news coverage on spiders and spider bites. It is overall coverage of the world. So this presentation will also presented by Angela Zhang on tomorrow. So thank you.
Thank you very much. We have plenty of time for questions, if anybody has questions. We have a question from Prashant. He says, thank you. Could you tell us about barcoding efforts ongoing in India for other arachnid groups?
In arachnid groups, this is first time I have worked in the spiders in my area. So because there are very less people only working on spiders, but in my area in Tamilnadu, myself and John Caleb also working on the molecular group, but he is working on jumping spiders, myself working on wolf spiders.
And is there work on other groups other than spiders?
No, sir. Not yet. Okay.
Any other questions for Dr. Dharmaraj? Now we have some people saying great job, and they're glad you're working on arachnid diversity in India because there's not many people doing that. So thank you for your work.
Thank you, sir. Thank you very much.